So aerobic respiration. For aerobic respiration to take place within mitochondria, the final products pyruvate is transported from cytoplasm into the mitochondria. The crucial events in aerobic respiration, we are completely oxidizing it, leaving three molecules of carbon dioxide, and the electrons removed as a part of hydrogen atoms to molecular oxygen for so that you get what? So that you get H2O. It's simultaneous synthesis of ATP. What is interesting to note is that the first process, okay, well, I'll tell you another one thing now itself. The reason why this aerobic continuation is good, another one's just on an additional point. This NADH is there now. You are able to regenerate NAD plus with the help of oxygen that we'll see in electron transport chain. What is interesting to note is the first process takes place in the matrix of mitochondria. Matrix of mitochondria, while the second process is located in the inner membrane. What are they talking about? Here they're talking about Krebs cycle. Here they are talking about, what are they talking about? They are talking about ETS, inner membrane of mitochondria. Okay, we'll remove all the unnecessary stuff so that we don't have any piling, it doesn't look dirty or unhygienic to look at. Pyruvate, which is formed after glycolytic catabolism. It is formed by glycolytic catabolism in the cytoplasm after it enters mitochondrial matrix. So where is this reaction taking place? In mitochondrial matrix, undergo oxidative decarboxylation. Why do we call oxidative? Hydrogen is removed. Oxidative decarboxylation. Carbon dioxide is removed by a set of reactions catalyzed by pyruvic dehydrogenase. You are removing hydrogen, dehydrogenase. Very simple. Logically itself you can remember. The reactions catalyzed by pyruvic dehydrogenase require the participation of several coenzymes. What are these several coenzymes? This is also very important. You need NAD plus, so you're able to get NADH. You need coenzyme A. You need Mg2 plus. We already have pyruvate dehydrogenase. So what are the things you need? This is 1, this is 2, this is 3, this is 4, this is 5. 5 prerequisites. Okay, wait a minute, my bad. This is not several coenzymes. We also need another thing. We'll see about that another thing in the extra portions. We need NAD plus, we need coenzyme A, we need Mg plus 2, we need pyruvic dehydrogenase. Okay. During this process, two molecules of NADH are produced from metabolism of two molecules of pyruvic acid. The acetyl CoA, this funny little guy who just has two carbon, enters a cyclic pathway. And what is this called? Tricarboxylic acid cycle. Okay, more commonly called the Krebs cycle after Hans Krebs. What is this guy? Who's this guy? Who's the guy we're talking about? Hans Krebs, who first explained this. So the tricarboxylic cycle kind of starts with the condensation of acetyl CoA, acetyl group, with oxaloacetic acid OAA and water to yield citric acid, a 6-carbon compound. The reaction is catalyzed by citrate synthesis. You're synthesizing citric acid, yes. So citrate synthesis simple, and a molecule of coenzyme A. That coenzyme A, that's it. It's gone. It's just sent away. The citrate is then isomerized to isocitrate. It is then followed by two successive steps of decarboxylation, leading to the form of alpha ketoglutaric acid and then succinyl CoA. Like first decarboxylation from 6 carbon to 5 carbon, and the 5 carbon thing is alpha ketoglutaric acid, and then we get succinyl CoA. First we'll see the theory and then we'll see the cycle. In remaining steps, citric acid cycle, succinyl CoA. We have a CoA, the CoA came back, interesting. During is oxidized to OAA during the cycle so that the cycle can continue. During the conversion of succinyl CoA to succinic acid, 1 GTP is produced. Succinyl CoA to succinic acid. Very important stuff. These are very important. Succinyl CoA to succinic acid is where GTP is produced. When we come to the cycle, we'll know, we'll see where and all, in which steps which is produced. But this you have to remember very much because this is something people often make mistakes. They confuse this succinyl, this with something to happen here with something which usually happens here. They confuse succinic acid dehydrogenase reaction with this reaction. So please be very clear. Succinyl CoA, this coenzyme A thing into acid, GTP is produced. Like GTP when it's produced, it's simultaneously, like you can utilize GTP to produce an ATP too. So you can say ATP or GTP, whichever you feel comfortable. But when you have a selection, when you have to choose something, choose GTP. This is a substrate level phosphorylation. What is substrate level phosphorylation? Any process in which ATP or GTP is produced without ETS in substrate level itself, when within substrate thing itself, when it's phosphorylized, when ADP, adenosine diphosphate, is phosphorylated, it is called a substrate level phosphorylation. In a coupled reaction, GTP is converted into GDP with the simultaneous synthesis of ATP from ADP. Okay, this is what I told. Okay, I've told this. Also, there are 3 points in the cycle where NAD plus 3 points. 3 points where NAD plus is reduced to NAD plus, and 1 point where FAD plus is reduced to FADH plus FADH2. And this 1 point is the succinic acid dehydrogenase thing. We'll see about that. The continued oxidation of acetyl-CoA via the TCA requires the continued replacement of oxaloacetic acid. That's just like how in how photosynthesis you need regeneration of RUBP. Every cyclic process you need the regeneration of the starting material. In addition, it requires the regeneration of NAD plus and FAD plus from NADH and FADH2. So regeneration of oxaloacetic acid is simple. We are studying in Krebs cycle itself. But regeneration of NAD plus and FAD plus we'll be studying in where will we be studying? We'll be studying in this ETS. The summary equation for this has been written down. So one pyruvic acid gives 4 NADH and one FADH and 1 ATP. So till now we have seen that glucose has been broken down to release carbon dioxide and 8 molecules of NADH and 2 molecules of FADH. 8 molecules of NADH and 2 molecules of FADH have been synthesized besides the 2 molecules of ATP in TCA cycle, tricarboxylic acid cycle. You may be wondering why we have been why we have been discussing respiration at all. Neither oxygen has come into picture, nor ATP has come. And this whole thing, once this connecting step is ETS, you are going to utilize. What are you going to utilize? You are going to utilize this oxygen, and at the same time, you will be getting a lot of ATP too. We'll see ETS next. Yes, so now we'll be starting electron transport chain. So we'll first break down what this is. What is electron transport chain? We have electrons, yes, associated with hydrogen. We are transporting them from one carrier to the other carrier, okay. And at the same time, we are doing an oxidative phosphorylation. What are we doing here? Oxidative. And we are, because of this oxidation process, we are phosphorylating an ATP. What are we oxidizing? We are oxidizing hydrogen protons, and we'll be oxidizing protons. We'll see about that. And electron transport chain. When we are transporting the electrons, we are getting energy from each of the redox reaction, each of the transports. So now let's get into the NCERT lines. The following steps in the respiratory processes are to release and utilize energy stored in NADH, assimilatory power, NADH and NH plus and FADH2. Note this point, when I say assimilatory power, don't take it literally. I'm just telling it to make you familiarize with the NADH and FADH. When anyone says in assimilatory power, you have to think about NADPH only, because that is what NCERT has, or that is what it has been given, and it is given for photosynthesis. Okay, continuing, this is accomplished when they are oxidized through electron transport chain and electrons are passed on to oxygen. So this is where oxygen comes. Oxygen is essential for removing the electrons, resulting in the formation of water. The metabolic pathway through which the electron passes from one carrier to another is called electron transport system. So here, electron transport system, if they ask if there is a situation, you have to remember that although it is a set of molecules, it's a set of carriers, it is essentially a pathway. So it is essentially a pathway. You can adjust it and take its meaning as some other thing, like the different systems, complexes. But essentially, what do they mean? They mean it is as a pathway. Just like how electrocardiogram is the paper is the result, and electrocardiograph is the machine. Electron transport system is a pathway. It's a metabolic pathway, and it is present in the inner mitochondrial membrane. Electrons from the NADH produced in the mitochondrial matrix during the citric acid cycle. But remember this, not only citric acid cycles NADH, but also the NADH produced from the glycolysis are oxidized by the NADH dehydrogenase that is, what is this NADH dehydrogenase? It is the complex one. So this whole system, we have a lot of complexes. Different complexes have different functions. We'll get to it. So we have complex one, which is sodium, which is NADH dehydrogenase, and then we have complex 2, which is succinate dehydrogenase, and then we have complex 3, which is cytochrome BC1, and we have cytochrome 4, which is cytochrome C oxidase. Okay, so 1 minute. Yeah, my bad. And electrons are, now what happens is these electrons from the complex 1 are transferred to ubiquinone. This is very important. 1 minute. Ubiquinone is very important. And where is it? It is located within the inner membrane. It is located within the membrane. See, it is present inside the membrane, not in the left, not in the right. It is present inside the membrane. Please pay attention to these stuff. Ubiquinone also receives reducing equivalence via FADH2, complex 2, just like how NADH has been oxidized to NAD plus, and the hydrogens from that after passing on to FMN and FES. What are FMN and FES? Pyridoxine S proteins. These are also intermediate electron carriers, electron transporters, if I may. And in this process, from the hydrogen, electrons are separated and the protons are accumulated in the intermembrane space. Now the electrons. We have seen that ubiquinone receives. Now ubiquinone. What is known? Known means double bond O. After it takes hydrogen, it becomes alcohol. Ubiquinol. I don't think this is an NCRT, but remember this. Okay. Wait a minute, I'll just write it here. This becomes ubiquinol, located within the inner membrane. Ubiquinone also receives reducing equivalence via FADH2 that is generated during oxidation of, yes, succinic acid. We saw succinyl CoA to succinic acid releases GTP or ATP, and now the succinic acid is oxidized, and that oxidation, I think it is oxidized into fumaric acid. Succinic acid or succinate is oxidized into fumarate, and the enzyme is succinate dehydrogenase, and that enzyme itself is complex 2. Understand this very clearly. The reduced ubiquinone, okay, they have given ubiquinol, is then oxidized. How is it oxidized? That is, it releases its hydrogen. Whatever it took, it releases. How is it oxidized? With the transfer of electrons to cytochrome C. So the next cytochrome it takes it is cytochrome C via how BC1 complex. So if you see the diagram, the electrons are going to cytochrome BC1 complex B, and then C1, and then C. Now we'll be seeing about cytochrome C. Yes. Now cytochrome C is a small protein. First important thing which you have to know is about the structure, is about the constituent. It is a small protein, and it is attached to the outer surface of the inner membrane and acts as a mobile carrier. What does it carry between the BC1 and between complex 4? So 3 and 4. Complex 4 refers to the cytochrome C oxidase complex. Complex 4, what is complex 4? You are having cytochrome C, so you're oxidizing that. So complex 4 is cytochrome C oxidase. What does it contain? If you see the diagram, everything you can get it from the diagram. So you have cytochrome A, you have cytochrome A3, and then you have some copper centers. Pardon me for the lower quality of pictures from the NCRT over here, but you will be having more clearer from your book. So please refer to that. Okay. And two copper centers. This two is also important. This also have been asked in exams. When the electrons pass from one carrier to another via complex 1 to 4 in electron transport chain, they are coupled to ATP synthase. ATP synthase is our complex 5. We'll see about complex 5 in the next coming in shortly. for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depend upon the nature of the electron donor. NADH gives rise to 3 ATP. FADH2 gives rise to 2 ATP. This is always true. I'm going to give you an extra information, that is the NADH which is produced in glycolysis. In glycolysis can there will be a popular statement that NADH of glycolysis can give rise to either 3 ATP or 2 ATP. Now we have a contradiction. But actually there is no contradiction. It is NADH when it's being transported into the mitochondrial matrix. If it is transported as NADH itself, then 3 ATP. But NADH can be converted into FADH2 also. And that happens in something called glycerol phosphate shuttle. This happens, when does this happen? Glycerol phosphate shuttle. We will be seeing about this whole conversion in the later part, in the respiratory, in this topic. I'll show you the topic in which we'll be seeing again respiratory balance sheet. In this, we'll be in detail. We'll come again to this topic. For now, we'll finish electron transport chain. Although aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage only. Where is it limited? Here we are removing with hydrogen and electrons. We are removing it as water. Only in the end stage we need oxygen, but even at that stage is very crucial. Now, presence is vital since it drives the whole process by removing the hydrogens from the system. Oxygen acts as a final hydrogen acceptor. Okay. Oxygen is the final hydrogen acceptor. Unlike photophosphorylation, here another one important thing you have to notice, hydrogen consists of protons and electrons. So in the electron transport chain, what is the final electron acceptor? That is also oxygen only. Okay. Unlike photophosphorylation, where it is the light energy that is utilized for production of proton gradient, you remember in photosynthesis, right? You'd have light, photolysis type takes place, water is split, and that is the main energy through which hydrogen is put inside, and hydrogen, because of the gradient, it comes out, and ATP is produced. There, photo-driven process. So photophosphorylation. Here, electrons are continuously undergoing redox reaction. They are being oxidized. So what is this? That is why it is called oxidative photophosphorylation. We have already studied about the mechanism of membrane-linked ATP synthesis as explained by chemiosmotic hypothesis in the photosynthesis chapter. As mentioned earlier, the energy released during the electron transport system. Energy released during electron transport system is utilized in synthesizing ATP with the help of ATP synthase. If you look at ATP synthase, we already know about the structure, but still I'll say again, this is the outer side, that is this is the inner we know that protons come from intermembranal space, and where do they come into? Inner membrane, right? So here the protons when they are going through, the first thing they come across is the F naught. F naught is embedded completely in the inner mitochondrial membrane, and after that we have the F1. F1 is the second part, and this is the main crucial part which helps in this process. And protons go through these are basically transmembrane channels. These are channels through which these protons are going to flow, and when they flow through these channels, they create an energy, and that energy is used to form ATP from ADP and inorganic phosphate. Why are we saying inorganic phosphate? Because like ADP, adenosine diphosphate, in that it is adenosine, so it already has that ribose sugar. Because of that sugar only, that is organic. Without the sugar, this is inorganic. Now I think it will be very clear why it is inorganic phosphate and that is just ADP. Okay, we'll continue. This complex consists of two major components, F1 and F0. The F1 is head piece, is a peripheral membrane protein. Very important. Okay, this is not that important, but it's good to know. Head piece is peripheral membrane protein complex and contains the site of ATP, synthesis of ATP from ADP and inorganic phosphate. F naught is integral membrane protein complex. Be familiarize yourself with the terminologies. Peripheral, it's in the outside membrane protein. Peripheral membrane protein complex. Integral, it is an integral part. It is inside that integral membrane protein complex that forms a channel through which protons cross the inner membrane. The passage of protons through the channel is coupled. Passage is coupled. This is how it forms. Coupled to the production of ATP. How is it coupled? It is coupled to the catalytic site of the F1 component. For each ATP produced, what happens? This is also very important. Two protons pass. How many protons? Two protons pass through F naught from the intermembrane space to the matrix down the electrochemical proton gradient. What is the gradient here? Electrochemical. Why is the word electro is there? Because there are electrons also involved here. I mean, there is positive ions and there is lesser positive ions. And why proton? Because we're talking about protons, electrochemical proton gradient. So the important parts of the chapter is now over.